Good afternoon, everyone. Thank you for joining us for this month's APN webinar. We're quite a few people already, and then the session is being recorded, so people will be able to watch what they missed at the beginning if they do join late. Today, I have the honor of introducing our speaker, Dr. Marelize Immelman. Dr. Marelize completed her undergraduate training at the University of Pretoria, where she also obtained her BSc honors degree in genetics. She subsequently moved to UCT, where she completed her master's and PhD theses in pharmacogenomics, supervised by Professor Colette Dandara. Dr. Immelman was a Vera Bradley Foundation scholar at Indiana University in the United States between 2016 and 2018, and has authored 20 peer-reviewed articles. She is a HPCSA registered medical biological scientist, currently working at the National Health Laboratory Service Human Genetics Laboratory at Groote Schuur Hospital. Her main responsibility is the Constitutional Microarray Diagnostic Service for identification, characterization, and reporting of genomic copy number, variants, and regions of homozygosity in pediatric patients with developmental delay / intellectual disability. Wow, a lot. Autism spectrum disorder and congenital anomalies. Her talk will cover the pharmacogenomic implications of microRNA regulation of genes affecting disposition of efavirenz and rifampicin. Thanks again for joining, and over to you, Doctor.
Thank you so much, Bianca, for the kind introduction, and good afternoon to everybody. Thank you for joining. I hope you can hear me well. And
indeed.
Let me just see if I can share my slides. Here we go. There we go. So the topic of today's talk is a bit of all data that I have sort of dusted off and just updated a bit, some of it. And the topic is microRNA regulation of genes affecting disposition of efavirenz and rifampicin. So efavirenz is a medication that was previously used as first-line HART as an antiretroviral therapy in South Africa. It was widely used until more recently when newer antiretroviral therapies became available. However, it is still used in some cases where some of the new therapies are counterindicated or cannot be used for some reason in a specific patient. So what I'm showing here in this slide is the metabolism of efavirenz, and it takes place primarily in the hepatocyte. And efavirenz is an active compound that is inactivated to mainly 8-hydroxyefavirenz, and this process happens primarily by the enzyme CYP2B6, but there's also a minor contribution by CYP1A2 and CYP2A6, CYP3A, and also UGTs. So the UGT2B7 is the main responsible enzyme for producing efavirenz in glucuronide. But there's also with efavirenz an interesting process that's called autoinduction. And how that works is efavirenz is able to activate two nuclear receptors. So they are called PXR and CAR. And they can in turn activate the CYP2B6 enzyme again, or CYP3A4 enzyme, and that can increase the rate of metabolism of efavirenz. So it's not only the drug metabolizing enzymes that play a role in the metabolism, but also these nuclear receptors that contribute. Sorry, let me just see why the slide is not moving forward.
click on the slide itself and then try moving it.
It's just saying wait for program to respond. Sorry. Maybe I must just try stop sharing and sharing again quickly.
Yeah, and reshare, yeah, I think.
Because now it looks like it is stuck, unfortunately.
You can, in the meantime, maybe just, if you
That, let me just...
send to me, then I can also see if I can.
It's opening it again. Let me just quickly share again in the meeting. There we go. Is it back again?
Yeah,
Okay,
we have
excellent.
it.
Excellent. Sorry about that, everybody. Let me just go back again. Okay.
Yeah.
So when we then look at rifampicin, this is a medication that is used for treatment of TB in South Africa, and it's not metabolized by the same enzymes. However, it is also known to be able to induce some of the cytochrome P450. So, for example, CYP2B6, CYP3A4, UTT2B7, the drug transporter ABCB1, and also SLCO1B1. And because it's often used together with efavirenz in patients that have both TB and HIV, it is important to study these enzymes and to look at what the combined effects of these drugs can potentially be when taken together. So a list of the relevant genes I've given here. So some are from the cytochrome P450 superfamily, others are phase 2 drug metabolizing enzyme genes, and then another is a transporter, so the ABCB1 gene codes for the P glycoprotein protein or drug transporter. And then, as I mentioned, the two nuclear receptors, PXR and CAR, that control the metabolism of efavirenz. So as a group, we have extensively published and tested in terms of pharmacogenomics associations with efavirenz over many years. So we have looked mainly at the role of genetic variants in CYP2B6, but also in ABCB1 and in PXR and CAR as well. And we looked mainly at the known variants that are known to affect efavirenz metabolism and efavirenz plasma concentrations. And all of that is sort of summarized in this table, where we saw that the main significant variants that contribute to different median efavirenz plasma concentrations include the ones from CYP2B6. So the 4 I have shown here are the significant contributors in this list of different variants. And the list continues, but these other variants were not significantly associated in our South African HIV/AIDS cohort. But there has also been other studies across the world that have looked at CYP2B6 specifically in various populations across the world, and that has resulted in a CPIC guideline being developed for how to use CYP2B6 genotyping when prescribing efavirenz containing antiretroviral therapy. So this is available on the CPIC website, and it can be used for prescribing efavirenz. So what is interesting is when you look at the CYP2B6 activity, and this figure from a paper from 2022 shows that even if you group CYP2B6 activity for certain liver tissue donors into poor metabolism, intermediate metabolism, and extensive metabolism, there's still quite a large degree of variability between individuals. So again, in this diagram here on the side, you can see although the extensive metabolism group has a higher expression of sub-2b6 mRNA in liver tissue, there's still a degree of variability within each group. So also for the intermediate metabolizers and the poor metabolizers. So we are obviously interested in understanding why is there such a range of activity in different people. So because we had already looked at the known variants, we were then concerned with the rest of the gene. So the gene also has other regions, for example, the 5' UTR, the promoter, the 3' UTR. And we know that those other gene regions are important as well, in addition to the known variants in the exons. So that's why we then wanted to focus on gene expression regulation. So gene expression regulation is a process. So mRNA is produced and then exported out of the nuclease. And then it is involved in translation, which then forms the protein. But obviously, there are certain factors like RNA stability that plays a role in how much protein is being produced and what a level of activity is observed by the enzyme. Oh no, don't be stuck again. There we go. So there's another mechanism as well through which gene expression can be regulated. And this is called, like I mentioned, the PXR and CAR nuclear receptors. So these nuclear receptors are able to bind to certain regions just upstream of the drug metabolizing enzyme genes. And by binding and interacting with these gene regions, they can increase the expression or decrease the expression of a particular gene. So this is very interesting when we look at efavirenz, because we know that PXR and CAR can bind upstream to, for example, sub-2b6, and that can then affect the level of expression of sub-2b6 and the rate of efavirenz metabolism. But we were not only interested in that, we were also interested in the regulation that is caused by short RNAs. Is the slide moving? Can you see the slide again? No?
Yes.
Oh, sorry about that. I don't know why it stops responding. So short RNAs are produced in the nucleus as well. So they are formed first as pre-miRNAs, and then they undergo processing by, for example, DROSHA or DGCR8 into pre-microRNAs, and then that is exported into the cytoplasm, where they are further processed by an enzyme called dicer, and they can interact with mRNA, in particularly the 3' UTR, to repress mRNA or to result in cleavage. They are able to bind to certain sites. So there are canonical sites that are known, and then there are atypical sites, and there are also marginal sites where they interact with a specific mRNA. And there's another thing that's called MERS SNPs, and that is genetic variants that are either in the 3' UTR of the mRNA that the microRNA interacts with, or that are in the region of the genome that produces the microRNA. So we were particularly interested in exploring these different variants and how they could potentially affect the regulation of the mRNA products. So that was our one paper, is where we looked at in silico prediction of microRNA targeting. So we focused on a certain set of genes and we looked at genetic variation in the 3' UTR and to see if they are able to potentially change the binding of certain microRNAs. So we know that we have the 3' UTR lengths for some of these genes, and we looked at how many SNPs are known to occur in this region and how many microRNAs are predicted by in silico prediction tools to target each of the different 3' UTRs. We used 15 different programs and we looked at how much overlap there are between the certain programs. So the percentage is the number of the same microRNAs picked up by at least 2 algorithms. And I've mentioned some of the algorithms here below in the table. And then we went in further to look at, for these particular microRNAs predicted to target a certain 3' UTR, we looked at whether or not they are also expressed in liver tissue. So the ones with a hash sign next to it are the ones that are also expressed in liver tissue. So they are of interest as well. And then we went further to look for genetic variants that are in the 3' UTR. So this table on the side here lists the ones that we were able to find when we sequenced the entire 3' UTRs. But I'm just showing a few examples here on the left-hand side. We also found some variants that have not previously been reported in DBSNP at that time, and they are also listed here in the table. And then we try to see if those genetic variants could potentially be predicted to affect the binding of a certain microRNA. So then we looked at whether or not the presence of the variant can change abolish, or create a target site for a particular microRNA. So the ones listed here are the ones that are likely to cause that effect and could have an impact on the control of mRNA expression. So then we wanted to further see if Efavirenz and Rifampicin actually has an impact on mRNA and microRNA expression. And we choose to use HepaRG cells as a model cell line for livers, for hepatocytes. And we then expose these cells to Efavirenz and Rifampicin in vitro and then measured the mRNA and microRNA expression levels. So this is a figure showing the effect of Efavirenz and Rifampicin on mRNA expression in the cell lines. So when you look at this plot, you can sort of clearly see that a significant change in sub-2b6 fold change for mRNA levels is being caused by rifampicin. And for Efavirenz, it's causing a significant increase in sub-3A4 levels, and then rifampicin also causes a massive increase in sub-3A4 mRNA expression. We're also just showing the DMSO, which is the solvent used to dissolve these Efavirenz and rifampicin in, and that is just a control to show that that doesn't cause any change in mRNA expression. But there's also the sub-3A5 that are significantly changed by Efavirenz, as well as UGT1A1. And then, interestingly, the expression of the nuclear receptor CAR is quite decreased by both Efavirenz and rifampicin. So when we then look at the microRNA expression, for this we use small RNA sequencing, and we wanted to identify which microRNAs have altered expression following exposure to the different medications. So overall, we had 10 samples that were done, and we had different numbers of reads that mapped to the regions in the genome for each sample. And we also have different numbers of microRNAs that are identified in each of the samples. So the ones with the highest expression is given here on the right-hand side of the table. And yeah, so that's interesting. So what the 10 different samples are is we did it in replicates as well. And then we looked at which microRNAs are affected the most. So the plot on the left-hand side is for efavirenz, and the plot on the right-hand side is for rifampicin. And you can see, for example, the microRNA 548D is significantly upregulated or higher expression following efavirenz treatment in this particular cell line. And then an example of rifampicin causing a decrease in microRNA expression is for this microRNA 876. It followed that sort of significant decrease following rifampicin exposure in the cells. So then we went further because we wanted to see, do we actually find some of these 3' UTR variants in our South African HIV/AIDS patients? And does it potentially affect efavirenz response?
You are
in this
muted,
group of
Mari.
patients. So we in SIP182, SIP2B6, UGT, 2B7, and also. Can you hear me now?
We can hear you now.
Okay, sorry. So we did genotyping of the 3' UTR variants in our South African HIV/AIDS patient cohort. We looked specifically at variants for SIP182, SIP2B6, UGT, 2B7, and also the NR1I2 gene, which codes for PXR, the nuclear receptor. And we use a variety of different genotyping methods. Hello?
Sorry, I'm not sure if it's just my side, but I can't see any slides anymore.
Oh my goodness. And let me just reshare again. Can you see it now?
Yes, thanks.
Oh, okay, perfect. I don't know what happened. So we also use snapshot genotyping and also sequencing for one of these variants to be able to detect it in our patient population. And we had quite a large number of patients that we could test for, so ranging from about 200 to about 350. And then we looked at the genotype and the allele frequencies for all of these different variants in our HIV/AIDS population. And some of them are quite common and some are more rare in the particular population. But this provides data on these 3' UTR variants in South Africa, which are not well studied. And so this contributes to that kind of information on South African patients. We also compared our South African patients to the frequencies in other populations. So, for example, other African populations, European populations, East Asian populations, and American groups. And the ones in bold is where we, as South Africans in this study, differ from the other populations. So you can see clearly there are quite a lot of differences with the other populations. And then we went on to look at these variants in terms of the efavirenz concentrations that we had, steady state efavirenz concentrations for these patients, to see if any of these variants are significantly associated with increased or decreased efavirenz concentrations. And the main one that we're finding is this SIP2B6 C1421 T2C change, and although it's not completely significantly associated, there is a bit of a decrease in efavirenz median concentrations with the individuals that have the C allele. But that is the only one out of those that were almost significantly associated. We also did a regression model where we wanted to see if we combine the effect of some of these variants with the previous known variants that do affect efavirenz metabolism, if we end up still seeing an effect of the other variants or if it's overshadowed by the effect of the known variants. And as you can see here, the main contributors are the CYP2B65162T variant and also the 983T2C variant. And those are the two well-known variants causing altered efavirenz metabolism in South Africans. Then after I completed this particular PhD study, I then had the opportunity, as mentioned, to spend time in the US, and there I was part of a certain study who looked at CYP2B6 UTR and if it alters the in vitro and in vivo activity of CYP2B6. So we then looked at certain variants in CYP2B6 and how common they are in that particular population. But these are ones that are also seen in our South African population, and they have quite a similar minor allele frequency, actually. And then we went further to look at the enzyme activity for CYP2B6 and to see if some of these variants in the 3' UTR actively change the enzyme activity. And we can see that for some of these variants, they do in fact result in a difference in enzyme activity, as shown here by figure A, B, E as well, and some decrease in activity for the variant studied in figure F. And then we wanted to get a further idea of how they affect the protein function or further measure of enzyme activity. So bupropion is used as a measure of CYP2B6 enzyme activity. And we can see that, again, some of the variants do decrease. If the variant is present, it does result in a decreased level of enzyme activity. And when you look at the difference in sequence caused by the variant, that could actually create a better binding site or disrupt a binding site for a particular microRNA. So for example, here in figure A, if the AG is present in the mRNA, then it is more difficult for the microRNA 625 to interact with the mRNA. Whereas if the CA is present, then there is actual interaction with some interaction with the GA. And when you look at another example, so microRNA 1275, it can affect or it can bind directly with the CA being present, but not with the AG in the mRNA being present. So we also looked at the expression of these microRNAs in certain cell models to see how they change the expression. So this is the ratio of the wild type versus variant, and some of these microRNAs are actually quite highly expressed, they kind of change the enzyme activity quite a lot. And the same with this study when we're looking at Firefly-Ranella ratio. They do, the fact that there's the variant can alter the ratio of expression. And so then a further study by the same group looked at whether or not microRNAs can be used as biomarkers for CYP2B6 enzyme activity. And they looked at the expression of these various microRNAs, and you can actually see that some of them, the expression is significantly different when you compare with the presence in liver tissue. So just to conclude, we were able to have a better understanding of the variability in treatment response. We looked at the role of microRNAs specifically, but we also looked at potential variants that can affect the binding or interaction of microRNAs. And then we looked at whether or not efavirenz and rifampicin has an impact on mRNA and microRNA expression. So we do see that the HepaRG cells is quite a handy cell model for liver cells. And there is an overlap in microRNAs with altered expression and between the two drugs. So that could be a potential contribution to drug-drug interactions. And there is a value in looking at both in silico testing as well as in vitro testing and also population studies. But there is obviously a high degree of genetic diversity and some population-specific changes when it comes to these variants that might affect microRNA binding. And I would like to acknowledge the team at UCT, as well as specifically human genetics and also clinical pharmacology, and then the team in the US that also did part of this work. Our Professor McPhail and Dr. Takuva, who initially collected many of the HIV/AIDS patients, and then also the Central Analytical Facility at Stellenbosch University that assisted with the short RNA sequencing, as well as various other funding bodies. So, for example, the Cape Town Carnegie Corporation, who gave funding, as well as the Medical Research Council and also UCT. So thank you for listening, and I'm happy to take any questions.
Thank you so much, Dr. Marelize. And this very informative talk. And also, I can see that a lot of hard work has gone into that. So congratulations on all the outputs and knowledge provided to especially me as well. I've been using quite a lot of data from your manuscript, so thank you for that. I'll open the floor now to any questions. You can raise your hand, unmute yourself if you have any questions, or just type it into the chat box. Maybe I can ask something then while people are thinking.
I'm trying to figure out how to raise my hand. I don't see anywhere.
Yeah, it's weird. I also don't see it.
Let me see. Okay. Oh, I see it now. Sorry.
Go ahead, Prof.
Okay. Thank you, Marelize, for this tour de force of the work that you have done. My question to you is where to now in terms of how do we take this work forward? Well, what are your thoughts on that? Thank you.
Yeah, so that's a great question. So I think that there still needs to be some additional work that now looks at, now that we're starting to understand that the genes are also regulated by other things like, for example, the microRNAs, still needs to be a little bit of work to just get a completely clear handle on that. For some of the, for example, the CYP2B6 is the most best studied example, but then for the other enzymes, potentially there can be some similar studies like some of the ones I've shown that looks at activity and also interaction with actual microRNAs. And then we have to figure out what exactly is the contribution of these changes in the to the response and the efavirenz levels, because if it's just going to be minor changes, then that might not be clinically so significant. So then there needs to still be a little bit more work, I think, on figuring out exactly how that impacts in the kind of clinical sphere where patients are actually treated. Is it at the same level of impact that one needs to actually test for these particular variants, or is it not necessary?
Thank you.
Anyone else with a question? I'm wondering what your opinion is on the use of other drugs, for example, like rifampicin, that has been shown to enhance the expression of these enzymes, to use that in the clinical practice to enhance patient response to, for example, treatment of a specific drug when they have a variant that causes decrease in function of said enzyme.
Yeah, so theoretically that should be something that one can consider doing, but then obviously one would have to do proper studies where you treat patients with both and you control for the dose and you measure exactly how that impacts the metabolism or the concentrations in the blood. So one would need to have those specific patients based on their genotype potentially, and then exposed differently in terms of drug treatments when they might have, had different dosage that they were on. So that would kind of involve a whole clinical trial situation, probably where you would control for the genotype and have your comparison groups and then see how the response differs. But yeah, because the only other way of answering that would then obviously be in cell models, but then you limit it still in terms of it's a cell model and you don't quite still know the impact in a general clinical setting. So yeah.
Thank you. Are there any other questions? If not, I would like to thank Dr. Marelize once again for her wonderful talk. And then there was a flyer shared in the chat. So I would like to ask Nicole to maybe, if she's willing to speak a bit more about that flyer and the summer school that's being hosted soon.
Thank you, Marelize. Let's wait for Nicole and then Nosipo for housekeeping. Thank you so much.
Yes,
Thank you for
good
having me.
afternoon, everyone. Thank you so much for the opportunity, and well done on your great research that you are doing. So I'm Nicole. I'm a lecturer at the School of Pharmacy at the University of the Western Cape. I am a pharmacist by training. I'm currently also busy with my PhD. So for my PhD, I want to develop a curriculum framework for formation in South Africa. And I would like to invite pharmacists that are experts in the field to participate in my study. I would also like to have some local experts. So everything is in the flyer. If you have any questions, you're welcome to reach out to me. And then we are also hosting pharmacogenomics winter school and a stakeholder engagement workshop in July in Pretoria, but it will be offered hybrid as well. So anyone that would like to learn more about pharmacogenomics, you're very welcome to join us. And then we would also like to invite anyone that are representing any university or any society to also participate. We need everyone to make pharmacogenomics implementation a success in South Africa. So you're also welcome to reach out to me as well. And we can share more information via.
Thank you. Nosipo, do you have any housekeeping, any issues that you want to highlight?
I have nothing, Prof. Thank you so much, everyone, for joining. Our next webinar is going to be on the 3rd of July.
Thank you. Much appreciated.
Thank you so much for having me.
All right, with that, that's the end of this webinar. Thank you everyone for joining. Oh wait, I have a
Thank
hand.
you, Bianca, for
Was there a
sharing.
hand? Dr. Lisa, or was that just a goodbye
Okay.
wave? Okay, thank you everyone.
Bye.
Thank you. Bye.
Bye-bye.